Good morning. So we have lots to get through today, and I want to talk about the assignments and the midterms. So let's get right into this. First thing, I just wanted to remind everybody that the assignment is due 1 week from today, so that's November 10th. And so I know I've had a lot of questions about it, and hopefully everyone's working hard on it and going to do wonderful. So just a reminder about what the assignment is about, if you're still working on it, is that you're going to be choosing a microbe from the topic list, and you're going to be making a web page or a fact sheet about it, and you're going to have all of the following headings. So do make sure you do follow the headings. It makes it easier for me to mark and easier for you to get the good marks, and that you do follow the instructions that are written under each of the sections. I think I talked about it at the beginning of the semester. I'm not going to give you a lot more details unless you have any particular questions on this. What I did want to talk a little bit about was the grading and the referencing, because I had a lot of questions about that. You can see this is the grading rubric I made, and kind of depending on where you fall, you can get 0 to 2 marks for each section, or 0 to 4 marks for the presentation. So do make sure, like I said, you follow the instructions, and you'll get good grades on this. So let's just talk about the referencing for a little bit, because I think there's a lot of people that want to reference this like an essay, which is kind of okay, but it's not really what I'm looking for. Okay, I do applaud your desire to make sure everything is cited properly and all that, but this is kind of a little bit of a different assignment. And so you can see I have this note here, and it says, choose 3 good resources for someone looking for more information. Okay, so I am assuming that probably a very high percentage of the material you're going to find is going to fall under what I'm going to call common knowledge. And I'm not worried about citations, because this isn't an essay, it's a different type of kind of assignment. What I'm looking for are 3 resources. So I want you to give references of things that are really good reads. So that if, let's say, this particular project I'm showing you right now was done on tuberculosis, and I'm really interested and I want to find more about tuberculosis, I want you to give me 3 good resources for that. And that's what I'm looking for. So 2 of them, or sorry, 1 of them has to be some sort of book or journal article or something like that, and you can use your textbook. That's perfectly acceptable, assuming the textbook does have good information in it. Some of the diseases are not in the textbook. So some people are having problems getting into the meeting. Okay, yeah, so thanks for that. Yeah, I had some issues too getting into it at the start. So hopefully this will resolve itself. I don't know whether this is a Zoom issue or a Kano internet issue. This last week has not been good for me for computer issues. I will just say that it's been a bit of a nightmare. But anyway, so hopefully people can get in. And if you're having serious issues, take a look at the recording later that I post on the YouTube list. So like I said, I'm looking for 3 good resources. You can do up to 5. I don't want to see every sentence cited like an essay and all that. If you're doing a fact page, you're not going to have a lot of space. You're going to find that there's this is different than an essay, and that you're trying to find ways to kind of be concise, yet provide me with good information around your assignment. You have a little bit more flexibility with a web page because you're not restricted to 2 pages, but you're not going to have a lot of room if you're citing everything using APA style or something like that. So what kind of information do you need to have? If you have a website, I do not just want the URL, I want the author and the title. So the author may be the Lung Association or the Public Health Agency of Canada or something like that, and the title may just be tuberculosis or the name of your disease, but that's what I want to see. And then the URL. If you're citing a book or a journal, you probably know that we're looking for more complete bibliographical information. So the year of publication. For journals, you've got volume and page numbers and other publishing information. I'm not looking for necessarily a standard format, but it'd be good if you were consistent in using something that looks recognizable. but I'm not going to be looking for any for particular formats. I'm looking for complete information is what I'm looking for. Your images are probably not going to be your own. I'm imagining most of you don't have gram stains or photographs of things like tuberculosis or whatever at home. So I'm imagining you're going to get those from internet sources. And so just putting URL with the source is sufficient. Do try to, if you find an image source, you know, if you're getting it from a blog or something, often they have taken it from someone else. So try to go to the main source if you can, and give the actual person credit who took it. So if you get an image from Wikipedia, for example, take a look at the Wikipedia credits, and you'll see they're often getting their images from the CDC or you know, different government agencies is where they got a lot of other medical images. So the other thing I just wanted to note here is I'm showing you this one for a couple of reasons, because this person here apparently got her image from Google. Google is not, you don't reference Google. Google is a search engine. Okay, go to the source. So that's not what you should be doing. I was also going to point out with her other references. So this was good. She had some information, lung association, but there was probably a title to that article. That was missing there. Same thing with the one on the top. It looks like you've got the organization, but not the title. So hopefully that clears up a few things for everyone. Just a reminder that if you have not done so, I think probably 3/4 of the class has done so. They've given me their plagiarism certificate. If you've not done so, it just has to come in with the assignment. And if you hand it in with the assignment, that's fine, but I won't grade your assignment until I have this. So if you haven't done that, you can just upload it with your assignment when you hand that in, and that's not a problem. Okay, so I do want to talk about the midterm. I know everyone has been anxious about it, and it feels like it was so long ago. I have some things to say about the midterm. Let me just pull up a PowerPoint here. And I gotta share it. So let's talk about the midterm too. I was hoping to be done today, but as I mentioned, I had some computer issues, which shut down my computer for a few hours yesterday, having IT looking at it. So I'm a little bit behind. I think I've got about, there's 50 something people in this class, and I think I've got about 15 more to grade. So I might be able to finish it today. I do have a lab this afternoon that I need to work on for another class, and I'm not sure how long that's going to take. And but in theory today, guaranteed by Thursday. So I would recommend maybe check the grades 5 o'clock today. If that's not done, there's a possibility I might bring it home this weekend or this evening. But I will guarantee by Thursday. But you know, please be patient. You know, I think everyone's been bogged down this year, myself included. I tell people this year's been like working a new job. I'm learning so many new things, and I'm doing everything differently. And a lot of things are taking longer than anticipated. But I'm getting there. So I will get there very soon. Like I said, Thursday at the latest, hopefully today by 5 o'clock, but I'm not going to make any promises on just in case I don't deliver. So I do want to kind of go over the midterm a little bit because I know some people had some questions. So I just wanted to kind of overview some of the questions that were giving people some problems. So just a reminder of where you're hoping to get is hopefully a C+ or better. So when your grades come through, hopefully that will give you an idea of how well you're doing. And I think I have Moodle programmed so that it should automatically give you a letter grade based on this scheme. So I will make sure that's the case when I do update the midterm grades. So I just want to take a look at some of the questions, not all the questions, but some of the questions that I think I was looking at questions that more than 75, less than 75% or less than 70% of people got correct. So these are the tougher ones apparently. You can see the first one, Helicobacter pylori. This is the one that lives in the stomach and sometimes gives people ulcers. And so ulcers are not in the large intestine, they're in the stomach. Not usually classified as a superbug. So the right answer is resistant to acidic pH. Which of the following does not describe a method used to culture viruses? Growing the virus on a petri dish containing nutrient broth. So viruses can't eat stuff. They can't eat food. So that's the correct answer there. I think a number of people picked this one here, human volunteers. And I know I did mention in class that with some viruses, that's our only way to culture them. We don't have options. So for example, norovirus, we cannot culture without using human volunteers. So yeah, lots of fun for those people. Good thing about norovirus, it may give you violent diarrhea and vomiting, but it rarely kills anybody. Okay, a question about remdesivir. And so, yeah, it looks like this is correct. It's been used to treat Donald Trump for getting SARS-CoV-2. It does act by inhibiting a viral RNA polymerase. It is a prodrug, meaning that the original chemical is actually converted in the body to the active drug. It was originally developed to treat Ebola, and it is most effective when it is taken when people actually have the virus. So that means the first maybe 4 days of infection. After that, the virus load in the blood goes down, and the symptoms are kind of collateral damage where the immune system is overreacting and all that, and it's not really going to be that effective. That seems to be the case with a lot of antivirals for acute infections. Okay, this is one of the matching questions. I just lined everything up with the correct answer. I can't remember how people did on the matching questions, but I was able to, these were easy to cut and paste just into the PowerPoint. So hopefully people did well on this one. I'll just let you look at that for a moment. There are 2 serological kind of tests. You've got the immunofluorescence. So you're visualizing something. ELISA is where you're detecting antibodies or antigens. Okay. Drug type with mechanism of action. Again, like I said, I can't remember how people did on this one, but it was easy for me to cut and paste these. So here there are the answers. I'll just let you take a look at that for a moment. Okay, a couple more. Antibacterial drugs. So basically just matching the drug with some sort of description. So erythromycin is something that attaches to ribosomes, and it's often used when people are allergic to penicillin because it's using a completely different mechanism of action. Tetracyclines are strong broad spectrums. Fluoroquinones, refers to DNA synthesis polymyxins found in topical skin treatments like polymyxin or polysporin. Okay, let's talk a little bit about the written questions. These had a lot of variability in terms of what people were giving me. So you can see this first question was about why gram-positive cells don't appear pink. So some people proceeded to write out the entire gram stain procedure. I did not need that. I needed an explanation as to why they turn purple and not pink, right? So kind of the bottom line was that they're stained purple, and the purple is a stronger, darker color, and so the red doesn't show up over that. So that's what I was looking for. He did write out the entire gram stain procedure, but didn't really answer the question. I gave you half a mark. List 2 uses for PCR. So these were this is right out of the notes. Some people said fingerprinting. Fingerprinting is where, people have fingerprints, right? Like we've got a little bit of oil on your finger and it ends up on a crime scene and they dust it and add ink and all that. So just a reminder to make sure you get your terminology right. The technology is called DNA fingerprinting, and that is used for forensics and paternity testing. So, just to make sure that you get that correct. Some people were really giving me the bare minimum. I think there was somebody who wrote just identification. Well, you got to tell me what. Like, what are you trying to identify? Pathogens. Examples are great. Tuberculosis is a good one, or chlamydia is another good example. I thought that was amazing how few people actually gave me that as part of their answer. Is that, considering this class, we're trying to identify pathogens, right? So that was interesting. This one here, Why do I have these stars? This was maybe the most important question on the midterm in my mind. I mean, it was only worth 1 mark. I think most people got it correct, but a few people did not. So what is the correct answer here? The flu is caused by a virus, right? So that's kind of important information. That's why we don't give penicillin when people have the flu or a cold. And what does penicillin do? It works by inhibiting enzymes involved in peptidoglycan biosynthesis. Or you could say cell wall synthesis in bacteria. So like I said, very important question, and hopefully you got that right. There were a few people that had a couple of weird answers, but most people did do well on that one, so well done. Okay, so this one here, I'm asking to sterilize, so that means we're killing all the organisms on some surgical tools. And so this one here, you had to have autoclaving to get full marks, okay? Out of 5 marks, if you didn't get autoclaving, the best you could do was 3 out of 5. That was the best answer by far. And what I was looking for is, if you can read the description, you know, describe how this is accomplished, right? So autoclaving, you got to tell me it's an instrument that uses pressurized steam, and it will denature proteins and membranes, and you know, so information like that, right? Irradiation, UV irradiation was probably one of the other good bets there, and telling me that it damages DNA and whatnot. And ethylene oxide and dry heat were also probably the best ones. A lot of people had talked about chemical disinfectants, and if you had autoclaving, you could still get 5 marks on this. But for chemical disinfectants, it's not just good enough to list aldehydes and then tell me they cross-link proteins. You have to tell me how you're going to do it, right? You know, like I said before, your job is to convince me that you know what you're talking about. So soaking it in the disinfectant, yes, that would probably work. Depending on the disinfectant, you might have to soak it shorter or longer. Maybe it would have to be washed off afterwards if you're applying this to somebody who's having surgery or not wanting to introduce chemicals to their body. So you may have to rinse them in sterile water afterwards. But as long as it's soaking or something like that was good. Wiping it off is not good enough. That would not be sterilization, that would be more disinfecting. Some people gave me some weird answers, things that don't seem to work. You're not going to filter scissors or pasteurize forceps. So like I said, you know, looking for details and examples is better, right? Again, irradiation, some people talked about irradiation but didn't really tell me what kinds. So again, you gotta give me lots of good details for the full 5 marks. Okay, and then this one here, I don't think I have the answers written down here, but I think most people did okay on this question. Some people lost marks for not really being thorough again, right? So you can see, for example, question A, it says, describe what is observed on the blood agar. So a lot of people said beta hemolysis, which is correct, but I want more than that. I want you to tell me what it looks like, because that's what the question says. Describe what is observed. What does the technician see in the gram stain? Purple cells, not good enough. I want you to tell me that we're seeing streptococcus. So the purple cells are circles or spheres, and they're found in little chains. So like I said, just trying to be, you know, give me that full, you know, this is the difference between people that are going to get an A and a B and a C, right? Is you know, that they're giving me good, complete information and demonstrating that they have a thorough and complete understanding of everything. Okay, so I know people are wondering where we're at. So just a reminder, we are at 45% of the grade. So we've had 2 midterms and a quiz. We have an assignment next week. So I think last year the assignment average was something like 78%. So the assignment average is usually better than midterm averages. Some people can do very well on it. I think most people could probably do quite well on the assignment. But again, you have to you have to put in a little bit of work. It has to look nice. It has to be accurate. You want to make sure your grammar is good, all those kind of things, a nice polished report with good information. Do make sure you look at, like I said, look at the assignment sheet to make sure that you have complete information, okay? You know, I'm talking about, you know, for example, the classification section, right? The classification section, it really kind of depends on the kind of organism you have. But if you have a bacterium, then you want to tell me, is it gram-negative or gram-positive? Are there other details to know? E. coli is not just good enough to talk about E. coli, for example. There's different strains of E. coli. So if you're looking at a unique strain of E. coli, you know, it's a good idea to tell what's different about that strain than normal E. coli, right? So 0157, for example. So, you know, like I said, you want to be, look at those instructions, make sure you have good information, and, you know, and so that's a big part of it. Also, like I said, being polished and having good grammar and all that is of course, very important. And then lastly, there's the final exam. So I think we have a date for that. I don't know it off the top of my head, but the exam schedule is out. And so, you know, that will be the last chance for people to pull up their grades. And some people certainly do. So if you are in a risk situation, we can certainly talk to you about that. So somebody's saying December 10th at 9 a.m. Okay, that's correct. Someone says, I can't see the midterm on Moodle. No, I'm grading them. So you can't see them yet. And so we'll get those done soon. I know people are having some anxiety over the midterm, and like I said, I'm working hard to get it done, but just I've been bogged down and it's been happening a little slower than I had anticipated. Okay, I do want to move on, but I will answer this last question about the final exam. Final exam is worth 40%. So in terms of the length, it's kind of similar to 2 midterms. You have 2 hours and kind of similar structure, some multiple choice, some matching, some written, those kind of things. And it focuses on the last part of the course, but is inclusive, meaning it includes everything. So I would say about 70% of the material on the final is from the last few topics, and then maybe 30% from the first part. Somebody's asking if all the final exams are 3 hours. No, some are 2 and some are 3. For this course, I had been asked to follow the University of Alberta standards, and they have a 2-hour time slot. So that's usually what I do. Okay, so I would like to get back to epidemiology. We've spent enough time here talking about that stuff at the moment, and like I said, we'll get those midterms back to you as soon as I can get to it. I'm working hard to do it, and so I apologize that I've been slower than usual, but you know, this year has been, I think we just all have to be a little extra patient with everything, unfortunately, even if it wears at us. Okay, so epidemiology, I think we finished off talking about disease transmission, and we were talking about, in particular, vector-borne diseases. So we were talking about Lyme disease and West Nile virus, which are kind of the biggest, most significant vector-borne diseases in Canada. Lyme disease being transmitted by ticks, and West Nile disease being transmitted by mosquitoes. There are other vector-borne diseases in Canada, but the number of cases are like pretty small. Like Rocky Mountain spotted fever, I think the number of cases are like 10 or less. I could be wrong, but I know they're pretty small, and they tend to be in the Rocky Mountains, so west of them, in fact. So not too many cases of that. So if you are from BC, you may have heard of it. If you're not, you may not have heard of it. If you are from the United States or some warmer parts of this continent, then you may have heard of it as well. So I want to talk about a few other things regarding epidemiology. I want to talk about nosocomial infections. So nosocomial is kind of a funny word, and but it's used, and it kind of depends on where you go, right? I was looking at Public Health Agency of Canada, and they are using this other term, healthcare associated infection, where sometimes you see hospital acquired infection. And so you see in the literature a lot of people have HIA, or sometimes you might see HCIA. And like I said, you know, people love to throw around different terms, but it all really just means the same thing, right? It means that you have gotten an infection from some sort of hospital or healthcare facility, right? I think that's why we're getting away from hospital-acquired infection, because there are many types of healthcare facilities. And I think I had mentioned maybe about a month ago now that there's a number that's been floating around, and it's about 10%. So about 10% of people who have a stay in a hospital acquire some sort of secondary infection from that hospital stay. You can say I have a range there, 5 to 15, but the number is about 10%, and that number has been around for decades. We can do a little bit better. Some hospitals by design seem to be able to do a little bit better than others. You know, newer hospitals, we have, you know, better air circulation systems and whatnot. And sometimes, you know, hospitals get crowded, or, you know, they're not big enough for the population. And, you know, those can actually increase the numbers. But the number is about 10%. And it's just by the nature of the fact that we're in the hospital. This costs us a lot more, so that costs us a person usually an average of about, I think it's 9 or 10 days extra in the hospital on average, which is also not good. And some of these are life-threatening infections. So what factors are contributing to this? Probably some of them are obvious. You're in a hospital, people are sick, they have germs, they have pathogens, and you're more likely to get them there. This was a big deal in Italy with SARS-CoV-2. People were going to the hospital and then getting COVID-19 from other people that were sick. And they were talking about in Italy, unfortunately by design, for the last 10 or 20 years, they've been closing down all their small hospitals and making these massive large ones. And of course, when you have a pandemic, that's a great place for everyone else to get sick. So that's not good. Obviously, there's transmission between other patients and staff and medical equipment and whatnot. You can imagine, you know, if nurses or physicians are not washing their hands in between patients or clients, that can be a big deal, right? And so, you know, this is why hand washing is important. Can't emphasize that enough. Compromised immune systems, right? People are already sick. People are already, you know, you're looking at old people in long-term care, people that have other chronic conditions, and that can make them more susceptible to these kind of infections. And then kind of the last one, you know, the last sort of piece of this recipe, of course, is you've got all sorts of procedures that people are undergoing. In particular, we're thinking of catheters and surgeries and IVs, right? If they're not done sterilely, they can be good sources of infection for people. So I think somebody's asking about MRSA. Yes, MRSA is probably the #1 nosocomial infection. Kind of depends on the hospital. There's been so many interesting studies looking at these, and you can see there's a list of organisms here. And I was looking at the textbook, and I was trying to look at some research on this, and no one can necessarily agree on what the top organisms are, except for the 4 that I haven't read. So you can see Staphylococcus, Enterococci, Escherichia coli, and Pseudomonas aeruginosa. Those 4 seem to make every single list 100% of the time, and they seem to be the most important ones. Staphylococcus seems to almost always be #1. The order of the other 3 kind of depends. And you can imagine different hospitals are doing different things. Different healthcare settings may promote different things. Sometimes Clostridium difficile gets very close to being #1. In fact, with certain outbreaks, once in a while, it is #1 for a short amount of time, although Staphylococcus usually does stay at the top. So you can see on the left, it's talking about the different kinds of infections. They're probably pretty obvious, right? You know, you're giving people catheters, you're introducing something to their urethra, and it's very possible to get a urinary tract infection. You can see that's kind of the big #1 on there. And that's often E. coli, but many of the rest of these can cause urinary tract infections. You can see that's kind of a very brief, kind of limited list. If you look at what Staphylococcus can actually do, blood, urinary tract infections, and wounds, That's very, very small list. It can cause pneumonia. It can pretty much infect almost every single bodily system that you can imagine. So you can see some of the other infections listed on there. Bloodstream infections, respiratory tract infections. I noticed on that one that is, I can't remember where I got that data from. There's a similar one in the textbook and the numbers are slightly different. But now we're starting to see with Clostridium difficile, gastrointestinal infections kind of start to take a little part of that wedge, which is kind of, I think I mentioned before, this is kind of a newer thing. We're looking at maybe the past 10 to 15 years. And I don't really have an answer as to why Clostridium has really popped up so well in the last 10 to 15 years. Not that it wasn't present before, it just seems to be more and more important, more and more outbreaks. So what do? We wash our hands. It's the #1 thing people can do to control these things. Obviously, there's a bunch of other things if you think about all the good housekeeping that might be done in a hospital to prevent transmission. Medical devices, when they're going from one room to another, are going to be sterilized or disinfected. Some patients are going to be isolated in their own room. They're not going to have a roommate. They might even be in a special wing or a special room that is going to control for those infections, and that includes a lot of MRSA infections. Monitoring outbreaks, obviously, and effective hand washing. One thing I don't have on there is sometimes these guests that are isolated they might have visitors, and the visitors may have to wear gowns and gloves, and which are going to be removed before the person leaves. So I'm not going to say too much more about hand washing, other than it's super important. I could just flood you with tons and tons of studies on hand washing, you know, that's really #1. And you know, it's kind of like, just seems to always go back to what Mom says, you know, wash your hands and eat your vegetables. Some of the best advice you can get in life. So this is the CDC recommendations. This is from our textbook. I was going to actually look for a similar list through Public Health Canada or Alberta Health, and I found really long documents that were not nice and friendly. So maybe someday I'll make a nicer list. But you can see this is pretty basic stuff. And these are things that you'll be learning about if you're not learning about already in some of your nursing classes, right? About proper ways to handle patients aseptically, proper way to handle sharps and needles, you know, cleaning up bodily fluids, etc. Those kind of things. These are all potential sources of infecting, going from patient to patient. Okay, so kind of for the rest of the lecture, I want to talk about something called emerging infectious diseases. Sometimes we talk about re-emerging infectious diseases, meaning diseases that are coming back, but usually they're grouped in the same category. And so these are new things or increasing things or things that are kind of on the rise. And I want to talk a little bit about some examples and why these things may be taking place. So contributing factors, increasing population densities. So I mentioned the hospital in Italy, where suddenly it was getting full and a lot more people were getting sick. I'm kind of going to skip that one because I really want to talk about some of these other things that are more relevant to the last couple of years. So evolution of new strains. It's amazing, every year I update this list and there's different things on it. I think these are some of the more relevant things in the last 10 years. We've had 2 pandemics, an influenza H1N1 pandemic, and now the SARS-CoV-2 pandemic, and then some epidemics of some avian flu. So it's worth it to talk about these for a minute or two. So let's talk about H1N1. So probably most of you remember that to some degree, all the fuss that was happening in 2009 and 10. And so what happened there? So what happened was that we had several strains of flu floating around. So this was actually a quadruple reassortment. There were 2 swine flu viruses, 1 bird flu virus, and 1 human virus. And somehow in there, we don't know whether it was one event or a couple events, but all these viruses infected one organism, probably a pig, because pigs have all the right receptors to get infected by both bird and human strains. And the reassortment happened, and then we end up with a brand new virus that was brand new to humans, and it could infect humans and pigs. So that's kind of what happened there. And I think we talked about it a little bit already, and I mentioned that the good news was it was not as bad as the Spanish flu, where millions and millions of people died. Some people did die from this. Some people did get quite ill, but most people were okay. And we did have a vaccine for this within 6 months, which was also kind of nice. We had proven technology for flu vaccines. So we were able to nip this in the bud. That year, I believe something like 45% of Canadians got their flu shot. Normally it's, I think it's less than 25%. And the virus stopped in its tracks actually very quickly in Canada, which is really quite remarkable. So what about avian flus? I know you hear about these in the news sometimes, right? And with a lot of fear. And there's good reason why there's a lot of fear, right? So avian means bird. So hopefully you know that. What is going on with avian flus? Well, these are bird flu viruses. And the flu viruses in birds are actually not respiratory viruses. So my image is a little bit misleading. Bird flu viruses are actually found in the fecal matter. They're gastrointestinal viruses. So they're doing something different, but that means wherever you have birds, you have bird feces, and the viruses are there. And once in a while, these viruses can infect humans. And there's a whole bunch of strains that have been characterized over the years. You can see there's a few there, H5, H7 viruses. So I think I have one of them I want to talk about for a minute. And so what is going on here? So like I said, you can imagine that, you know, wherever there's birds, you could be a farmer, maybe you have chickens or ducks, and the birds could get that virus. And you know what, if you've ever had chickens or been to a chicken farm, you can see they are pretty messy animals. The feces is everywhere. And it's probably not that hard if you get some feces on your hands and contaminate your mouth. There's something that is done once in a while in these animals, they can fluff up their feathers and get the feces in the air. I was reading about, or a friend was telling me, where was she now? I think she was somewhere in Asia in one of those food markets, right? And you know, like we get our chicken, we go to the grocery store, it's all butchered for us. And she was telling me about how you go to this food market and there's the chickens sitting there in a cage, and you pick one out and the guy says, okay, and grabs it free, it's still alive, and he chops the head off in front of you and then hands it to you, and you bring it home, and you get to pluck the feathers and do all the hard work. And that was just the standard wherever she was. And I was asking her about one practice that I'd heard about that is pretty common. So a bird is worth money, right? And a lot of these people are not rich. So if one bird dies, that's a few dollars that you know, could have been in their pocket. So sometimes these birds, they choke on things. And you can give birds a type of rescue breathing where basically you put their beak up to your mouth and you suck on it and suck out whatever is in their throat. And so you can imagine there, the bird has something in its mouth, you're sucking and you're inhaling. And these people are actually pretty high risk of getting different germs from their animals because, of course, you know, they're inhaling things into the lungs. So this is pretty rare. Usually you're looking at a bird to human transmission, and usually these viruses are not transmitting human-human. And that's hopefully going to be the case for the future too, but you never know with these mutations, right? Here's the really scary thing, right? So this is H5N1 that we've been tracking for a number of years. Haven't heard too much of it in the news recently. There was that H7 strain that popped up. But we're tracking these. You can see these are cases in different parts of Asia. And take a look at that number I have up there. 700 cases and about 60% mortality. So this is terrifying, right? I mean, how many diseases do we know has that kind of mortality? Now, this probably is not the true mortality number, right? You're looking at relatively you know, poor people. Most of them are not going to hospital until they're, you know, quite ill. So there's probably a lot more cases than we're actually getting recorded. But let's say the cases are 1/10 of that. That's still 6%, which is pretty darn high. So there's a lot of people thinking, you know, and tracking this and hoping that it doesn't recombine with a human virus and make the next flu pandemic, which arguably was part of the case of what happened back in 2009. So like I said, we're keeping an eye on flu pandemics. And there's a lot that could be said about flu shots, and I'll talk about them when we get to our immune system lectures. So I will say a couple more things about this. This is, I believe, was it Korea, South Korea? I think it was. Anyway, they had the bird flu strain came through, and what do? They call all the chickens, so they kill them. All right, we're looking at, looks like 250,000 chickens. You can see there go the eggs. The concern is, of course, they're going to make everybody sick. So that's kind of the response, which is not good if you're a chicken farmer to lose all that revenue. So what about SARS-CoV-2? I had showed you this slide before. I was talking about coronaviruses, and it seems that coronaviruses historically have been jumping to humans for a long time. In fact, we didn't really know about these until the 1960s, and there's not been a ton of research on them because most of them, these 4 here in particular, are giving us basically mild colds for the most part. Most of these things seem to be coming from bats or rats, and often there's some sort of intermediate host. And I mentioned that, in the past 20 years, it seems like every decade we've seen another coronavirus. So 2002, we saw SARS Classic, and we believe that came from bats and went through a cat-like creature called a civet, and which is an intermediate host before it made it to humans. We've also seen MERS, which is Middle East Respiratory Syndrome, and that's been in Saudi Arabia and a few other areas in Asia. And these ones have been pretty highly pathogenic diseases. And then recently, so probably about a year ago or maybe more, we don't know exactly when it made the jump to humans, but we've got SARS-CoV-2. And there's not a lot of changes from, we found there was a specimen taken from bats. I can't remember what year it was. I think it was 2016 or something. That's something like 96% identical. So this is why we think it's coming from bats. And even more recently, we found an even more similar strain in something called a pangolin. So this is why we think a pangolin is the intermediate species before it jumped to humans. So some people are wondering what a pangolin is. I know I told you that before, that you need to look it up. I'll show you a picture. They are super cute. That is a pangolin. Where are my other pictures? I was going to show you some other pictures first. So let me just exit this for a second, because I know I had some other pictures, because they are just absolutely adorable. So pangolin is it's a type of mammal. They're really weird looking. So let me see here. I'll reshare that. So there we go. There's a pangolin. They're super cute. They're mammals with scales. The scales are very much like your fingernails. They're made of the same stuff like keratin. They're found in Asia and Africa. There's about 8 species, and I have another picture here. There's another picture, a little baby on the mom, and they're actually highly endangered. This is possibly the most trafficked animal in the world. They're prized for both their meat and their scales. There's a lot of traditional medicine that uses their scales, claiming it has all sorts of potent properties, but it's basically just like eating fingernails. I guess probably some of you who know if you chew your nails, you're not getting any extra benefits from that. They do curl up in a ball when they're feeling threatened, and I couldn't get enough of looking at photos of pangolins. They're certainly super adorable. So like I said, some people are thinking this was the intermediate host. I think there's some debate around that. Again, it's a virus that's slightly more similar to the bat virus, 97% or something like that. It's possible that the bat virus was able to infect humans and pangolins, and that there's some other intermediate host. But it doesn't really matter. The virus is here. It's in humans. It's not going to go away. It's probably not going to disappear. It's probably going to be with us for ever and ever. Anyway, that's what I think about SARS-CoV-2. So what other things are we talking about that can lead to new diseases or increasing diseases? We talked about antibiotics and how inappropriate use of antibiotics can lead to all sorts of resistant strains and superbugs. So the #1 superbug, of course, is Staphylococcus, and there it is. And I think we've talked about it quite a bit already, and how important it is for people to take this seriously. It can get into all sorts of wounds, and blood infections with drug-resistant bacteria are very, very serious. Lots of people die from this annually. So this is why we have special protocols for dealing with MRSA. It's just very good at infecting just about anything. It just needs that opportunity to get into the tissues, and hospital has lots of places to do that. There's a picture from the textbook just basically saying what I said already, is that these people are often isolated in caps and gowns, and you'll see signs. If you're ever in a hospital, sometimes you see signs about patients being isolated, and it's often MRSA. So like I said, Clostridium difficile is not necessarily the #1 superbug, but sometimes it pops up. Sometimes it makes a list of #1 once in a while. And it has a bunch of different names. I think I mentioned this before, right? This is the classical scientific name. Something that kind of happened recently is some of the literature is starting to use a new name. So I'm not really sure whether this has something to do with some taxonomy or some phylogeny. Somebody's looking at how related these things are, and but both names are still used. Most people are just calling it C. diff, which seems to cover both names. Once in a while, you'll see CDI as well, which is another abbreviation for Clostridium difficile. But for the most part, it carries all the same characteristics as other Clostridium organisms, meaning that it makes endospores and a few other things like that. So I think I mentioned before that this is often caused by, it can be part of your normal gut flora. It's not necessarily found in everybody, but sometimes people get antibiotic-induced diarrhea by getting some sort of other infection. They take antibiotics, it wipes out your normal flora, and you're stuck with this thing because it's drug resistant, and those endospores aren't killed by the drugs. And then it can cause diarrhea. All right, what else do we have on the list? Weather pattern changes. So Lyme disease, there's a lot of good evidence to show that it's moving north because our winters are getting a little milder. And as the winters get a little milder, the species that propagate Lyme disease, so white-footed mice and the ticks, are able to survive the winters. And so we'll see where that goes in the future. But definitely, if you look at where Lyme disease was, it was in the 1980s, it was found in one place in all of Canada. It was kind of one of the most southernmost places in Ontario. And since then, it's traveled and it's migrated and found in many of the provinces. What about modern transportation? So this is a good one, actually. I mentioned before SARS Classic in 2002 ended up in Toronto. One flight from Hong Kong, and it was across the ocean very, very quickly. If you look at the spread of the 2009 pandemic, how quickly it spread across the globe. We have airplanes everywhere. West Nile virus is a really fascinating story. This is a map I found, and it was originally characterized in New York City, and it spread across the continent. So it didn't arrive in Canada until the year 2000. It was in Eastern Canada, and by 2004, it had reached British Columbia. And so this was just, again, we don't know how it appeared in New York. There are some theories about exactly how it appeared in New York, and people think somehow it arrived on a flight. So I don't know whether that was an infected mosquito or what, but some people even think they know which flight it was, but I don't think we'll ever know for sure. But probably it had to do something with human transportation as well, which is, this is the modern world we live in, right? We get across the globe very quickly. We like to do that, but some of these things have consequences. All sorts of other things. You can see I have a list of things that have happened in the last number of years. We've had Ebola outbreaks, we had a cholera epidemic after the earthquake. If you go back far enough, some of these hurricanes, Hurricane Katrina, there were all sorts of diseases that were able to propagate very easily due to all of these issues, right? And so, you can imagine all the things going on, right? Water isn't getting treated. You've got populations moving and migrating, population densities. And so I'm not going to dwell on these for too long, but just some things to think about. So usually I ask students, what is this? And people are looking at it. Usually somebody gets it eventually. This is the globe, and this is a map showing flight paths all across the world. So if you kind of squint, you can maybe make out North America on the left, South America on the bottom. On the right, you can see that big bright spot is Europe and Africa below it. And of course, then I show people this map here and say, look at this. This is a bunch of emerging diseases that have been, I thought it had years on it, but I guess it doesn't, dates. And these things, they don't need passports. They move around, and if we can make things more convenient for them, they're definitely going to spread a lot quicker. Some people are terrified by this map, right? Disease has always been around, and the reality is in our modern world, we are healthier and better off than we've probably ever been. So we just have to take this and realize that we are in good shape, and use our knowledge and all that to combat these things, and hopefully we can do something about our current pandemic. Okay, so a couple other things to finish off this topic. If you ever go to like a travel clinic, sometimes people, they decide to go traveling, it's very interesting because you'll see maps that say, if you go here, you should get this vaccination and so on. And I saw this list here. And so there are some diseases you may or may not know about that are important in other geographical locations. This is a big one here, yellow fever, right? Oops, likes to do that on me. So yellow fever is not really a concern in Canada, but it's spread by insect in a lot of tropical locations. So there's some countries, if you travel to them, they want to have two things. They want to have your passport, and they want to have your record of yellow fever vaccination. And if you don't have that, then you get back on the plane. So that's something kind of interesting. Rabies, I thought this was really funny. Why would you get your rabies shot? It says, well, if you anticipate direct contact with wild carnivores. I'm not sure who fits into that category, but I found that kind of amusing because most people are not anticipating or hoping to have direct contact with wild carnivores. So I thought that was interesting. Somebody is asking a question about deliberately emerging. I don't know, is that on that map somewhere? I'm not sure what that's referring to. Deliberately emerging. Oh yeah, that's a good question. I don't know what that means. Huh. Oh, okay, yeah. I can see it's a black dot, and the black dot is looking at anthrax terrorism. So we're talking about terrorism or biological warfare, which is, of course, a threat. In some cases, thankfully, it's really rare. So all this requires records keeping. So you're looking at a huge number of organizations that do this. Alberta Health obviously keeps track of all the healthcare incidences in Alberta. Public Health Agency of Canada, there's worldwide places, World Health Organization, the CDC, they track disease worldwide. We don't track everything. People get colds all the time. We don't track them, but there is a list in Canada of notifiable diseases, they're called, right? This is a small part of that list. They fall into different categories. You can see some of the diseases fall into the category of things that are preventable by routine vaccination. We want to know how well our vaccination programs are going. Sexually transmitted diseases, waterborne, foodborne diseases, we want to keep track of, are there outbreaks in our food or our water and things like that, and bioterrorism. And usually anything serious as well, right? So things like Ebola, obviously we're tracking things like that, and right now we're tracking cases of SARS-CoV-2. I don't know if it's officially part of the notifiable list yet or not. I think there might be a legislation behind that. I'm not entirely sure. Some diseases, like Lyme disease, has only been notifiable since 2009. So it's been around since the 1980s in Canada, but we've only actually been tracking official numbers for 12 years. So here's some data. I was not able to find more recent data yet, but 2015. So you can see here's some cases here. So mumps, pertussis, and measles, and some Alberta numbers as well. And so pertussis, for example, is a good example of a disease where the vaccine wears off. And so some people still get it even though they've been vaccinated. Some of these other diseases are things where people were probably not vaccinated in the case of measles, because the measles vaccine is actually a really, really good vaccine. It's something like 98% effective, which is amazing for a vaccine. Sexually transmitted diseases and so on. So interesting to look at these numbers from year to year, particularly if you're tracking something like measles and you see the patterns there. So we'll talk about measles a little bit later on in the semester, kind of one of the last things we talk about, because of course it's directly related to vaccination practices. So some last thoughts on epidemiology. Okay, so here's something, right? And this is something we've been talking about this year. You know, how often do you touch your face, right? Especially in an era of pandemics. There's lots of good studies on this. You know, people touch the face. You scratch your eye, you pick your nose, or whatever, whether it's intentionally or unconsciously, and it happens all the time. And of course, this is where the pathogens tend to hang out. So most respiratory viruses survive 2 to 4 days, or can. This depends on all sorts of variety of things, whether there's mucus involved, the moisture, temperature, humidity, all those kind of things. So wash your hands, I guess. You don't want to handshake people, right? I guess no one's handshaking right now. So, you know, you can do fist bumps or elbow bumps, and you know, when you return, when you get back to the college, you know, and we're here in the hallways and you see me in the hallway, give me a fist bump and I'll know that you were listening to this lecture. I would really appreciate that. And I guess, yeah, I keep saying it, wash your hands. Hand sanitizers are great, but hand washing is the gold standard. Okay, so let me see where we are with time. We have about 20 minutes left, which is good, because I did want to break into the next topic and get into topic, what are we at, topic 12 now? Gotta make sure we don't get too far behind, but I think we are doing comfortably in terms of timing. We'll be ready for the final exam. I'm just going to get this PowerPoint up and we'll get going. You know, sometimes I like to go overtime because I'm just so excited to tell you about all this wonderful stuff, and hopefully you like it too. So next topic we're going to look at is, you can see the title. I've changed this title many times over the years. I think we're kind of getting to where we're at. It used to be called Infections and Disease or Health and Disease or something like that, because you know, our health, particularly regarding microorganisms, doesn't just have to do with disease. Some of them are actually doing good things for us as well. So this is something important to know, and we're going to talk all about that, and at least a little bit about it. So first, let's just think about our body, and this is something I've talked about before, is how our body is like an ecosystem. So if you think about the number of human cells on the human body, we've got about 30 trillion cells, kind of depending on how big you are. Some people are a little bigger, some people are a little smaller, somewhere on that order. We're looking at, and I know some people are probably lighter than 50 kilograms and some people more than 85, but just kind of an average range. And we think we've got about 20,000 or so genes in our genome. We probably have more bacteria than 30 trillion. So the recent estimates are saying that about 43% of the cells on our body are our own, and maybe the other 57% are bacterial. That represents about 3 million genes or more. Again, we don't really know. The number is massive. So sometimes people are talking about having our extra genome, talking about our microbiome. We'll talk about that in a moment. We don't know how many viruses are infecting those bacteria. Number is probably even larger than that. So there's a lot of things going on there. And think of it as an ecosystem is a very good way to kind of consider this. Is in ecosystems, you've got give and take going on. Some relationships are mutualistic, some are commensal, some are pathogenic. And you know, this is something that we are you know, just starting to barely understand. Super fascinating area. So somebody's asking about tuberculosis, classified as a parasite rather than a pathogen. You can use both words for tuberculosis. The word parasite means something that causes damage to your body. Often though, when we say parasite, we usually mean a worm or a protist, not usually tuberculosis, but it is technically a correct word. So we use these words, and we see them all over the place. And these words are used somewhat interchangeably: microbiome, microflora, microbiota. I think I put in your notes kind of some definitions there. There are slightly different meanings, but for all intents and purposes, they can be used interchangeably. So I remember back in the day, everyone used to use the word flora, and some people got mad because, well, the word flora usually means flowers and plants, and so then they came up with the word microbiota. And nowadays we're using the word microbiome. And like I said, it really is inspired by the genomics people who have been able to detect all sorts of species that are all around us. So we're looking at all sorts of different bacteria, but other organisms too: fungi and yeast, archaea, protists, and even some animals. And I'll show you one there in a few minutes. This microbiome has been connected to many, many different types of human conditions. So you can see there's a list there: Parkinson's, Alzheimer's, depression, obesity, inflammatory bowel disease. And like I said, we are just kind of barely starting to understand it. Really, really fascinating studies. Many of the studies have been done in rodents, so mice and rats. And of course, humans are way more complex, particularly when you're looking at things like depression. But there's some good indications that having a healthy microbiome is essential for good human health. So when we're talking about the microbiome, sometimes we talk about the resident flora or the transient flora. So resident flora, that means organisms that are with us pretty much all the time. So a good example of that is, of course, Staphylococcus epidermidis. It's on all of us, all of our skin. We get colonized when we're a baby, and it's with us for life. You know, unless you're undergoing some sort of crazy procedure where you're sanitizing your entire skin, you're not going to get rid of it. Transient flora are things that kind of come and go. So a good example of this is Staphylococcus aureus. Somewhere around a quarter to a third of us have Staphylococcus aureus on us. It can colonize us for weeks, months, or years, but you know, kind of it comes and goes as it pleases, I guess. I'm not exactly sure the mechanism behind that. Sometimes we get colonized for days, sometimes it's a lot longer than that. And so transient, like I said, kind of means it comes and goes. So these things are doing all sorts of good for us. And I think I mentioned this before, and there's a lot of good research behind this that we're kind of just beginning to understand. Some of these points will be touched on in future lectures as well. But I think I mentioned that a lot of these bacteria are in our guts, and they're making vitamins. So B vitamins and vitamin K, they can help us digest food. And believe it or not, that some of these organisms are essential for us to develop proper immune systems. So if you don't have the proper microbes when you're a baby, your immune system is not going to develop properly. Like I said, lots of really good research behind this, and we really don't understand a lot of it. We're just kind of slowly beginning to understand this. Obviously, the bad are the infections. We're talking about opportunistic infections in many cases. Some of them are producing toxins and waste products, and these things, of course, are bad for us too, and may cause some of the pathogenesis of the disease. Hope you like my little diagrams. I was looking on the internet. I was inspired by some little happy faces I saw. So like I said, this is something I can't get enough of lately. There's so many different studies that are popping out, and we're just like, we really don't understand what's going on. A lot of them are rodent studies, and so we're trying to figure out how we can do the human studies. Rodents are, of course, a lot easier to study than humans in terms of the ethical practices and whatnot. But lots of good studies. I was mentioning depression, right? There was a clinical researcher that had some clients that had been chronically depressed for a few years, and when he examined their fecal samples, he was finding a different profile in their fecal samples than he was finding in normal healthy people. And so he was saying something is going on here. So lots to worth talking about. And hopefully, we'll crack some of these answers in the next decade or two. So where do we get our microbiome from? The answer is everywhere. This kind of thing is studied using these germ-free animals that they can make. And what they do is to make a germ-free animal, the mouse is, you get a pregnant mouse and you clean her up and you treat her with a whole bunch of antibiotics and very sterilely you give her a C-section and you put those mice into these sterile chambers, and then you can study how microbes colonize those germ-free mice, right? So kind of one at a time or two at a time or whatever. And we've learned a lot from these procedures over the years. And we get our microbiome basically as soon as the water breaks when you're a fetus. There is actually some indication from some studies, they're a little bit controversial, that maybe some of the microbes are actually in the placenta while you're developing. But again, like I said, those are relatively controversial. So once your water breaks, you start getting bacteria introduced to you. And once you go through childbirth, you're getting bacteria from your mother's vagina. You're getting fecal bacteria from that process as well. And you're going to maybe feed from a bottle or a breast, and there's going to be bacteria on the skin of your mother, the skin of your father, and there's going to be there's bacteria all over the place. And depending on the species, some colonize right away. Some take a little bit of time to get into the system. When you switch from liquid food to other types of food, you start chewing your own food and all that. That's going to change the profile of your gut bacteria. And like I said, this process, some organisms take a couple of years, some take weeks, some are shorter than that. It's kind of a really interesting area of research. There's something else that's coming from this, because there's an obvious question here, right? What if you were not born vaginally? What if you're born through a C-section? And there's actually some pretty good studies on this. I found a good article. I think it was this was the BBC had a kind of an article referring to a study talking about so what's going on here? Is there a difference? And so in this study, they were able to find a few things, that there were differences in babies' bacteria that were observed. And the differences start to go away after a couple of years. But considering those differences are including the first couple of years of life, there's lots of questions. And in fact, there is some good data to show that children born by C-section are at higher risk of things like asthma and allergies, and apparently type 1 diabetes, which I didn't know until I read this article. It doesn't mean that we should stop doing C-sections. There's a good reason why people do C-sections. Sometimes there's danger to the birth of the baby or the mom, and it's a highly successful kind of procedure. I see someone has a question I was just about to address, and the question is about something called vaginal seeding. She says she read about people swabbing the vagina, wiping on the baby's faces. This was something that even a year ago in class I had said we don't know the answer to this, but since then there's been 2 or 3 studies looking at vaginal seeding, and they did not find a difference in this. And they're not sure. It's been proposed that probably some of the fecal bacteria are more important than some of the vaginal bacteria. But we're not going to start recommending that people smear their feces on their babies' faces at this time. But maybe in the future we'll find a good mix. We'll figure out what bacteria are really important for the baby, and the baby will get a probiotic or something like that. Again, this is very preliminary research. This has only been, people have been only trying it for a few years, and maybe future studies will show there's a certain technique or something like that that will be better. But at the moment, it's not recommended, but because there are risks, of course. The person could be giving their baby group B streptococcus, and we don't want to do that either. So what's going on naturally, and all these big questions? It's amazing how much we don't really know. Other than the bacteria are good. They're good for our immune system and all sorts of other things. So there's, like I said, tons of studies done on these kind of things. You can see that, you know, when you're a fetus, the number of bacteria you have is probably going to be near zero. And once you're born, you're going to start to get exposed to those bacteria. And as you start eating different foods, you can definitely see different types of bacteria between like vegans and meatitarians, people who eat spicy food. Generally, you have similar bacteria to the people you live with. And we see, you know, different profiles cross-culturally. So what do we know about this? The biggest thing to know is that diversity is really good. If you have a really diverse microbiome, that seems to be the healthiest. You can kind of think of it as like a rainforest, right? You know, you've got a nice forest there with lots of different species, lots of different plants and animals. Same thing with your microbiome. If you've got a lot of diversity, that tends to be really, really good for you. And people who have health issues, so like I said, those chronically depressed people, their diversity was hugely decreased. So this is like, okay, we don't know what's going on. This is a chicken or the egg thing too. Are they depressed because they have bad bacteria, or do they have bad bacteria because they are depressed, right? And these are very hard to tease out in humans. And like I said, depression research is really, really complicated stuff. So I will tell you one secret, or maybe it's not a secret, okay? People's ears are perking. I said the word secret. One of the secrets to having a good gut microbiome is eating fruits and vegetables and fiber. So I know just everything in life seems to come back to getting fiber. And, you know, I'm sure as we all get older, we're going to want more and more fiber. But yeah, that's a big secret, is that helps. It helps the good microbes grow, and it helps to have diversity. They like to feed on the fiber, and they like to live on it, and so that's a good thing to have. So like I said, eat your fruits and vegetables. That's a good thing. Okay, so a few minutes left. I kind of just want to talk about some of the microbes, where they live and what they are, and you're going to see a lot of familiar names and some that are probably not so familiar. As I've implied again and again, we have lots of bacteria in our gut. The majority of them, that's where we're going to find them, but we do have them many other places: on our skin, vagina, nostrils, mouth, all sorts of other places. So it's worth kind of talking about some of these and thinking about some of the potential pathogens that are there as well. So let's talk about the skin. We've talked about the skin already a little bit. The skin is dry and a little bit salty because we have sweat and other secretions. And so that actually inhibits a lot of things. In fact, it inhibits a lot of gram-negatives. And so we've talked a little bit already about, you know, our big two, right? Staphylococcus epidermidis and Staphylococcus aureus. Those are found all over the place. Most of us have, all of us have epidermidis, and some of us have aureus. Yeast, right? So there's a non-bacterial species. Candida is found on our skin and lots of other body places as just kind of a normal part of our natural flora. Now the conjunctiva, that kind of means the surface of the eye, and staph can be found there as well. And sometimes people end up with conjunctivitis or pink eye, which is an infection of that area. So let's talk about Staphylococcus for a minute. Like I said, these tend to really be fine with salty conditions, which makes them grow really well. There's Staphylococcus epidermidis, Staphylococcus aureus, and I have one new one to introduce you to, Staphylococcus hominis. This one was only discovered a few years ago. I can't remember what year that was, but less than 10 years ago. And so, you know, another Staphylococcus. How exciting? Well, maybe not so, because it turns out this one here seems to be related to body odor. Yay, or not so yay. Yeah, so for the most part, it seems kind of harmless, but what it does is it eats some of the oily molecules that are secreted from our skin and turns them into things that smell. So, you know, I'm not sure whether some people who have bad BO are more colonized with this or not, but I know that this organism is one that is connected to body odor, so not so fun. What other microbes are on the skin? I wanted to introduce you to this one here. I mentioned that there are some animals that are part of our flora. So how about eyelash mites? Okay, so just warn you, right? These kind of things, you know, the first time I learned about them, my eyes felt itchy. But I've gotten over this. I've gotten over it. I certainly have. These things are very tiny, okay? They are barely visible with the human eye. And there are other mites that live on the human body as well. I just found some good pictures of this one. And I'll zoom in there and you can see those little green things there, right? And this is kind of what they look like. As far as we know, I don't think they're actually causing us any issues. I think some people had concerns about allergies and whatnot. And as far as I know, they are not causing us any problems. And it turns out that the older you get, the more likely you are to get colonized by these things. I guess it's just a matter of time, you know, as we interact with people that have them. So I don't know a ton about them, but yeah, they're gross. And I try not to think about them as much as I can. But maybe now, you know, unfortunately, probably for you people, you will never unlearn this. So somebody's asking about fake eyelashes and mascara. I have no idea. I have no idea. I don't know much about these at all. You know, maybe if you learn something about these, let me know. Like I said, I don't think they're causing us any harm, but it's possible there are people with allergies. I think I read about one study where they're able to get rid of them by using some sort of chemical. But again, it's around your eyes, and it doesn't sound pleasant either. Okay, what about the other parts of the body? We've got the upper respiratory system. So mouth, nose, and throat. We're talking about maybe 1,000 or so different species. Again, you can see some common species here. This is kind of just some common ones that are kind of relevant to what we've been talking about. By the way, these are the same as the tables in the textbook, but what I've done is I've put the species that we're talking about in this course in bold. But if you are interested in the other species, you can certainly learn about them on your own time. But yeah, you can see there's Staphylococcus again, Staphylococcus streptococcus. Okay, so Staphylococcus streptococcus, Neisseria. We're usually talking about Neisseria meningitidis here because that can be spread by saliva. And so you're going to see on these lists, we're going to see the same organisms again and again. Staphylococcus, it's everywhere. It can infect everywhere. The different Streptococcus, kind of same thing. And then other organisms tend to be kind of a little bit more specialized locations. So potential pathogens, hopefully that one's obvious. Staphylococcus, Streptococcus, and Candida, which is can cause thrush, right? So we talked about thrush already as a yeast infection of the mouth. And of course, we also have vaginal yeast infections, which again, this is just something that's part of your normal flora, but when given that opportunity, it can overgrow. And some of the opportunities I think I mentioned before is when people have antibiotics. Some women tend to, you know, get a lot more yeast infections because, of course, it's wiping out the normal bacteria, and this is not a bacteria, so it has a chance to flourish. Okay, so almost out of time here. Maybe I'll just finish talking about the lower digestive system, and then we'll finish for the day. There are many, many, many species here again. Probably at least a couple thousand that can colonize us. There's about 200 or 300 that are kind of on the typical list. But like I said, you can imagine, you go all to different parts of the world. We have all different types of diets and those kind of things. And so you can see there's the usual suspects again. We've got Staphylococcus. We've talked about Enterococcus just a tiny bit. We've got E. coli, of course, is famous for living in the intestine. And you can see we've got Clostridium there, Candida again, and Helicobacter, and a few others. So these ones here, some of these on this list, you can see these are more, considered to be pathogens, so Entamoeba and Trichomonas. Okay, so I think just thinking about this, it's probably worth it for me to stop here and not talk a little bit more about digestive health. I know some people have some questions there about things like probiotics and you know, those kind of things, and we are going to get to that. It will just have to wait for next time. But there's things like probiotics and prebiotics and antibiotics, so we got all those things to talk about. But definitely we'll come back to this and we'll talk a little bit more about gut health next day. So hopefully your Tuesday is wonderful, and I will see you on Thursday. And like I said, you can check at the end of the day to see if I have the midterm grades. And if I'm able to get to it, I will certainly be able to finish that today, but I promise it'll be done by Thursday, hopefully a lot sooner. So have a wonderful day, and we'll see you on Thursday.